Hernan. I'm a research scientist here at Nemours, and I'm head of the Dynamics in Epilepsy and Cognitive Development Lab. I'm also affiliated with the Psych and Brain Sciences Department at the University of Delaware. And today I'm sort of going to talk about a little bit about my IMBRI pilot project and some data that led into the pilot, and just kind of tell you where we're up to with it. Okay, so epilepsy. So my lab studies epilepsy. So we're interested in this disorder. It's the third most common neurological disorder, with roughly 1 in 26 people developing epilepsy sometime in their life. And the incidence of epilepsy is not sort of equally distributed across the lifespan. So children are at a particularly high risk of developing epilepsy. And in these kids, about 50 to 65% of them will have some sort of comorbid cognitive or behavioral deficit. So that looks like, either something like a diagnosed DSM-4 disorder. So things like autism spectrum disorder, ADHD. This is developmental coordination disorder, depression or anxiety. And these are significantly higher than, the general pediatric population. So importantly, these comorbidities themselves are actually a pretty big predictor of quality of life for the patients and for their families. And, there are these bidirectional relationships with kids with autism in particular are often diagnosed with epilepsy, and kids with epilepsy are often diagnosed with autism, sort of suggesting something at the level of the mechanism that might be responsible for these cognitive impairments in these kids. So when you look at epilepsy as sort of a network disease or a neural network disease, these comorbidities make sense because there's always some sort of underlying etiology or developmental insult that leads to abnormal neural network development. And that leads to both these seizures as well as, sort of abnormal activity in between seizures. But given that the brain is functioning all the time, an abnormal or hyperexcitable neural network will probably process information a little bit differently. And this difference in information processing can lead to cognitive impairments, even, at times between the seizures. So standard therapeutic interventions often target these seizures. They do a great job in about 2/3 of patients, but the cognitive impairments often remain. So our goal is to identify a novel therapeutic intervention that would target some aspect of this abnormal neural network, and therefore be predicted to improve both seizure and cognitive outcome for these patients. So sort of for historical reasons, the drug that we chose to start with was ACTH. So ACTH is a hormonal treatment. It's an endogenous part of the HPA axis. It's actually a first-line treatment given to patients for severe infantile epilepsy. And there's some data in humans to suggest that this treatment is actually potentially better for long-term cognitive outcome in humans. And these data are, difficult to kind of parse apart because, they're epidemiological, they're in humans. So we actually have data in 2 animal models, in both a rat and a mouse, to show that ACTH prevents cognitive impairments in 3 different epilepsy models in 2 different species, and has actions above and beyond just a straight up corticosteroid. So this led us to the hypothesis that these receptors in the brain, melanocortin-4 receptors, are responsible, at least in part, for some of the mechanism for ACTH's effectiveness in preventing cognitive impairments after early life seizures. So these receptors are kind of spread out throughout the brain. They're located both on neuronal populations as well as glial populations, and in particular, astrocytes. And in neurons, they modulate LTP and other kind of neural network processes. And in glia, they are known to modulate neuroinflammation. So these are some behavioral data from an early life seizure model. Mohammed in my lab, who's now, I guess, a 4th year graduate student in the interdisciplinary neuroscience program at UD, he started using our mouse model and showed that basically similar to what we saw in rats, animals with a history of early life seizures, shown here in red, are able to acquire a fear acquisition task similar to their control counterparts, shown in blue. But when you ask them to extinguish a day later, when you ask them to learn to disassociate the tone from the shock in this task, so just sort of a standard learning task, the animals with early life seizures have a significant impairment compared to their control counterparts in blue here in this learning. And ACTH in green is effective at preventing this learning impairment. So these data we had published in rats before, but the mouse allowed us to then knock out the melanocortin-4 receptors. And what we found when we did that is, again, the animals acquire similarly, but the ACTH completely loses its effectiveness. So when we knock out these brain receptors, these neuropeptide melanocortin-4 receptors, we completely lose our ACTH treatment effect. So the seizures still cause an impairment, and the ACTH actually, if anything, is a little bit worse than the animals that were untreated and had seizures. So finally, this model, this particular transgenic mouse, allowed us to go and re-express the receptor in specific cell populations. So what we chose to do is re-express the receptor in neurons, and then chose to re-express the receptor in astrocytes. And all of these groups acquire similarly. But the interesting part is with the learning. So when we re-express in neurons, we do not recover our ACTH treatment effect. But when we re-express in the astrocyte populations, we do actually recover our ACTH treatment effect. So this was sort of our first piece of evidence, our first piece of really strong evidence that these melanocortin-4 receptors are really important for ACTH, and these are brain receptors, and B, that it may actually be the receptors potentially preferentially, or at least the astrocyte receptors are involved in the prevention of cognitive impairment that we see after early life seizures with our treatment with ACTH. So this was really exciting to us, and this sort of was what led to my IMBRI pilot project. So what are astrocytes? They are a large, diverse population of morphologically complex cells. They exist all throughout the nervous system, but they were previously thought as a glial cell, which literally means neuron glue. They were previously thought to be passive support cells for neurons. Progress in about the last 2 decades, I think, has shown that astrocytes are actually crucial for mediating development and physiological brain processes. They actively shape neuronal dynamics, so they're involved in things like oscillations, neuronal oscillations, and synchrony of large populations of neural networks. They're involved in structural and functional plasticity. They're involved in neurotransmitter uptake. So, ion balance, so maintaining, the proper level of potassium in the brain. So you can see how all of these things would be important for not just supporting neurons, but actually neuron function. But it's been a little bit difficult to test. So historically, the role of these cells as players in behavior has been called into question. But more recent data, again, indicates that they have sort of diverse mechanisms, and they can act at like the seconds timescale, behaviorally relevant timescales within neural circuits to shape behavioral outputs. So now we have some data that say that ACTH is, involved. ACTH is binding to these receptors on astrocytes, and that this is leading to a behavioral improvement. So what's going on with the astrocytes in our early life seizure model? So we wanted to look at this. We kind of hit a variety of these pathways that I just showed with just an immunohistochemical approach. So we looked at reactivity with a GFAP marker. We looked at glutamate clearance with GLT1 and GLAST. We looked at astrocyte to astrocyte communication with connexin 43. So I forgot to mention that these astrocytes are connected to each other, not only connected to neurons, but they're connected to each other with these gap junctions, these connexin 43 proteins, and they signal via calcium waves. So we looked at those connexin 43s to see if there, would potentially be a difference in how the astrocytes were communicating with each other after seizures, or the expression of the protein that allows them to communicate with each other after seizures. And then we looked at aquaporin-4, which is responsible for maintenance of ion concentration. So kind of hitting a lot of those major pathways that astrocytes are involved in. And so Manny in the lab was able to spearhead this. I should say also Sam, who was a really fantastic summer student and SERP student. And what they found was that after early life seizures, you get a significant increase in reactivity of these astrocytes and the GFAP expression that's modulated, that's mitigated or ameliorated by ACTH treatment. And you also get a decrease in the number of aquaporin 4 foci that's also mitigated by ACTH treatment. Interestingly, when we looked at GLAST and connexin 43, we don't see anything significantly different here. But there is sort of this trend in connexin 43 to actually have less connexin 43 in the seizure animals and even less potentially in the ACTH group. So we think that that's kind of interesting, but it's not significant. So this is in the prefrontal cortex. We also looked at the hippocampus. These two brain regions are really important for cognition. They're really important for that fear learning that I showed you before. So that's why we chose them. And we find the same trend in the hippocampus where, again, there's a significant increase in the reactivity of the astrocytes after seizures that's ameliorated by ACTH treatment. And then there's a change in aquaporin foci count that looks like it's actually kind of overcompensated for by the ACTH treatment. So our second aim in the pilot award was actually to look beyond sort of the static measure of what's going on in the long term after the early life seizures with the astrocytes. And we wanted to explore sort of dynamic changes in neuron to neuron communication, astrocyte to neuron communication, and astrocyte to astrocyte communication after early life seizures, testing the hypothesis that ACTH could potentially normalize any putative changes in an MC4R dependent manner. So this is actually some calcium imaging. That's a big neuron here in the foreground. You can see that it is actually dynamic, but the astrocytes in the background here have a very, very slow calcium transient. And we were planning on using an approach that we have previously used to measure neural dynamics to look at this communication between the astrocytes and the neurons. So we encountered a few snafus, but we're now tackling this in sort of a two-pronged approach with two different methods. So our first method is really to do these seizures, to treat with the ACTH. We're going to allow the mice to recover, and then we're going to take acute brain slices and either incubate with two different dyes that allow us to impregnate the cells with calcium indicator and label the astrocytes specifically. And then we're going to do some sliced calcium imaging. And those experiments are still ongoing. We've also we just got a really talented, fantastic postdoc who's interested in this question sort of broadly. And he's now piloting using a AAV approach to encode genetic calcium indicators into both astrocyte and neuron populations. And then he's right now, actually, as we speak, doing the surgery to do in vivo mini scope recording so that we, can actually ask the question of how these cells are communicating with each other while the animal is behaving and link that directly to behavior, both with the seizures and without the seizures in the context of treatment and without treatment. So sort of some conclusions, early life seizures are associated with cognitive impairments in patients, in mouse models and rat models. Cognitive impairments and seizures are secondary to any sort of neural network dysfunction. And these impairments can be prevented by ACTH, working through a mechanism that involves an MC4R agonism, potentially, like also specifically, I don't want to say specifically, but on astrocytes in the brain, probably on neurons as well, but on astrocytes, and that's what we're particularly interested in. And astrocytes are powerful modulators of neural network dynamics. We see changes in reactivity in both the PFC and the hippocampus that seem to occur after early life seizures and may be mitigated by these MC4R agonists. And we're currently exploring how these cell populations are talking to each other dynamically after early life seizures. So that's all I have for you guys today. My lab, I've got three wonderful graduate students. Mohammed and Akanksha Mohammed was doing a lot of the behavioral work and a lot of the in vivo neuron recordings. Akanksha is the one who's spearheading the sliced calcium imaging. That GIF of the culture calcium imaging is actually from a graduate student that I have at the University of Vermont who's defending on Friday. And his name's Trevor. Manny is a really wonderful research assistant in the lab, and he did a lot of the IHC. And John is our new postdoc who's doing the in vivo miniscope recordings. And thank you very much to the Delaware INBRE for helping fund this project.